Good evening, folks. I hope this finds everyone well. This is part 2 in my lecture series on Mendelian inheritance. If you have not yet watched part 1 on Mendelian inheritance, I might have called the lecture Heredity and Genetics or Heredity and Inheritance. I can't recall off the top of my head, but if you haven't watched it yet, you should probably go back and do that so that you have some examples of, so you know the foundations of what we're talking about. If you are in my biology class, you should get both the Punnett squares handout and the Mendelian inheritance handout from Blackboard so that you have all the terminology. You have some examples of Punnett squares in front of you. And whether or not you're in my biology class, I might recommend you have a pen and paper handy because we're going to be drawing some Punnett squares today, and I would like you to be able to follow along with them. Okay, so in the last lesson, we laid down the foundations of genetic science. We talked a little bit about Gregor Mendel and his research. We talked about the failed genetic theories of Jean-Baptiste Lamarck and Charles Darwin, who came before Mendel. Well, Darwin was contemporaneous with Mendel, just never read his work. And how Gregor Mendel, with his meticulous experiments on pea plants, worked out the fundamental rules of genetics. And among other things, last time we learned that each organism has 2 genes for each trait, which you know why that is, because you have paired chromosomes, one from mom, one from dad. Because you're inheriting 2 genes for each trait, that means that you have the possibility of getting 2 copies of the same gene if both parents sent on the same gene to you, which makes you homozygous for that trait. Or you could end up with 2 different genes at that locus, which means that you are heterozygous. If that happens, what happens to you? Well, the dominant gene is the one that gets expressed. Dominant genes are always expressed if they are present. Recessive genes are only expressed if no dominant gene is there to compete with them. And we have learned that an organism is going to, so in other words, you only express a recessive condition if you are homozygous recessive. If that's the only gene you have, you got it from both parents. And we have learned that the organism does not pass its traits on directly. It's passing on the genes, which is the instructions on how to build a trait. Ergo, that means the organism might not be expressing a recessive gene that it has, but it can pass it on to its kids because those genetic instructions are still there. So, with that in mind, I want to revisit Gregor Mendel's famous pea plant experiment. Last time I told you about one of the trials he did involving the color of flowers in the pea plants, that he took purple flowered pea plants, crossed them with white flowered pea plants. That was his F0 or starting generation. In the F1 generation, every single plant came out purple, and they were just as purple as their purple parent, no pale purple or intermediate color. Blending was right out the window. Then he allowed his purple flowered pea plants to self-pollinate. In the F2 generation, what he got out of that was 3/4 purple, 1/4 white. He allowed all of those plants to self-pollinate, and in the F3 and subsequent generations, he found that 1/4 of that 3/4 in the F2 generation that were purple was going to breed true for purple. They always made more purple when they self-pollinated. The remaining 2/4 or 1/2 of the population of the F2 generation repeated the F2 result when they self-pollinated, and the F3 generation onward, the white plants, when they self-pollinated, always made more white plants. They always bred true. So that was Mendel's results. Now let's apply what we have learned from this to figure out how the pattern of inheritance is working. Like, well, how do Mendel's rules apply to that experimental result? And I should like to point out an important thing about this as we go in, that Mendel's results were the averaged results of many, many, many experiments. You know, that he did not get perfect results the first trial or the 10th trial even, you know, but by averaging out the results of numerous experiments, the pattern did in fact emerge. As with anything in science, you never trust just one experiment. All right, though. So Mendel's F1 generation, or F0, his starting plants were a purple flowered pea plant and a white flowered pea plant. And we had already established that he had allowed them to self-pollinate for several consecutive generations, kicking out any plant of the wrong color that was born until he knew he was only getting purple and only getting white from the particular plants he was trying. In other words, they bred true. Mendel didn't know this when he started, but what he had created was a homozygous dominant purple plant and a homozygous recessive white plant. The purple plant had 2 copies of the dominant purple gene. The white plant had 2 copies of the recessive white gene. So what Mendel does is he sets up his experiment, he crosses them, and we want to see why he got the results that he got. There's a standard technique that we use in genetics for testing out the results of a possible crossing, basically to predict the possible outcomes of a crossing of two organisms for a particular trait that we are studying. And that is called a Punnett square, capital P-U-N-N-E-T-T, named after the English geneticist Reginald Punnett, who first developed the technique. Now, a Punnett square is a probability matrix. It's a little grid that shows the possible outcomes you could get from any given breeding experiments. Now, you can draw Punnett squares in different sizes, depending on how many traits you're tracking at once. But the most common ones you see, and the ones I'm going to ask you to be most conversant with, are going to be 4-box Punnett squares. Basically, you're tracking the inheritance pattern of one specific gene. Remember that every organism in your population has 2 copies of that gene, one inherited from each parent. Remember that Mendel's basic law of segregation tells us that it is random which gene is going to be passed on of the 2 that you have. So if I've got organism A, it's got 2 copies of its gene for whatever trait we're looking at. It could pass on either A or B. Doesn't matter which one. We got a second organism here. It's also got 2 copies of that gene. It could pass on either one. I'll spare you the math. 4 possible combinations of genes you could get there. So that's what the Punnett square shows us. Now, when we're setting up a Punnett square, a few things to know. First off, we use symbols to represent the genes in our study. Typically, we use capital and lowercase letters to represent those genes. A capital letter is used to represent a dominant gene. A lowercase letter is used to represent a recessive gene. Every organism's genotype can be written as a pair of letters. Remember genotype, G-E-N-O-T-Y-P-E. The genotype is telling us the genes the organism has. Whereas the phenotype, P-H-E-N-O-type, is telling us the actual visible trait being expressed. Now, any given organism has 3 possible genotypes. It could be homozygous dominant, in which case it has 2 copies of the dominant gene. We would give it 2 capital letters to represent its genotype. It could be heterozygous, in which case we give it 1 capital letter and 1 lowercase letter to represent the fact that it has 1 dominant gene and 1 recessive gene. Or we could have 2 lowercase letters to make it homozygous recessive. It has 2 copies of the recessive gene. So 2 little letters in that case. I should point out that for simplicity's sake, we are talking about a Punnett square in which there are only 2 genes available. There's 1 dominant and 1 recessive. In the real world, it's not always that simple. In the real world, the population may have 3 different genes for a given trait, or 8 different genes for a given trait, some of which might be dominant and some of which might be recessive. So in the real world, it can get a little bit more complex than this, but just to show you how it works, let's assume that there are only 2 options at that genetic locus, 1 dominant and 1 recessive. In the real world, it may be that you have 3 different dominant options and 1 recessive option, or 4 recessive options and 2 dominant options, or 7 dominant options and 0 recessive options, or 7 recessive options and 0 recessive options. There's no limit on how many alleles you can have floating around in the population for a given locus, and there's no guarantee that they're getting an equal number of dominant and recessives. But let's just say, for the sake of argument, that's what we're working with. Now, in Mendel's experiments, we saw that when he crossed purple flowers with white flowers in the F0 generation, all of his F1 plants came out purple. This strongly suggests to us that purple is probably the dominant color for flowers in peas. So we're going to assign it a capital letter to represent it. By the way, when you're picking letters for your Punnett squares, just a little tip to make your life more simple, pick capital and lowercase letters that are easily distinguishable from each other, so that you're not writing it down and somebody's saying, that one's a little bit smaller than the other one. Was it meant to be a little bit smaller? Is it actually a recessive, or just a dominant that you happen to draw a little bit too small, or a recessive you drew a little bit too big? So pick letters in which it's unambiguous which one is the capital and which one is the lowercase. I like R or D or G for that reason. Don't go with O or C or S. Now, other thing is that if you have a pair, if there are only 2 genes available for a given locus, it is standard practice in genetics to use the capital and lowercase of the same letter. Like if you've got 2 different genes available at that locus, then let's use capital R and little r, for example. Don't say, okay, we've got, green eyes and blue eyes, and we're going to make B for blue and G for green. The reason that can be confusing, because it's like, okay, if I made a capital B and a little g, if maybe we said blue was dominant and green was recessive, it's not, but let's just say we're doing that, then somebody might look at your results and say, okay, there's a big B and there's a little g. Do I know if there's actually a big G gene out there or a little B gene out there? It throws some ambiguity in about whether there are more than 2 available genes for that locus. So if there are only 2 possible options, we typically use capital and lowercase of the same letter. That's what I've done in my examples to keep things easy. Now, when you are drawing a Punnett square, here's what you do. You assign the genotypes of each of the organisms that you've got. For our example here, Mendel had purple flowered pea plant. We've determined that's dominant. We're going to say big R is the purple gene. That's the dominant gene. So we're going to assign it 2 big R's. It is homozygous dominant. We know that organism was breeding true. It was always passing on its parental trait when it self-pollinated. Ergo, that suggests that it's homozygous for it. So we're going to assign it 2 big R's for its genotype. Likewise, we know that the white plant was breeding true for white, that every time it self-pollinated, it produced more white flowered plants. We're going to assume it is homozygous for the recessive condition, and we're going to assign it 2 little r's for its genotype. So that is how we do this. Then we set up the Punnett square as a 4-box grid. Here is our Punnett square Can you see that one right there? There is our Punnett square for the F0 generation. What you do when you're setting the square up is you take one of the organisms, it does not matter which one, and you write its 2 genes across the top of the 4-box grid, 1 gene over each column. Then you take the other organism and you write its 2 genes beside the grid, 1 gene beside each row. Now the 4 boxes in the grid show you all the possible ways the genes of those 2 organisms could combine, because the top organism is going to pass on either its gene on the right or its gene on the left, whereas the side organism is going to pass on either the gene on the bottom or the gene on the top. Those are equally likely odds because, every organism passes on one of its 2 genes and they segregate randomly. So we have 4 possible combinations that can happen. So the upper rightmost box shows what happens if you pass on that gene and that gene. This box down here shows what happens if you pass on that one and that one, and so forth. Now you can see that for this particular Punnett square, the results are not terribly exciting because we have a homozygous dominant organism. It is going to pass on either a big R or another big R. That's its options. We have a homozygous recessive organism. It's going to pass on either its left little r or its right little r. In other words, there's no ambiguity about how this is going to come out. If I breed 2 homozygotes together, they're each going to pass on 1 copy of the gene that they have. Ergo, all the offspring, 100% of the offspring in the F1 generation, are going to be heterozygous. They all are guaranteed to get a purple gene from the one parent and a white gene from the other. Now remember, purple is dominant in this pairing. All these organisms have 1 dominant purple gene, 1 recessive white gene, and the entire point of being a dominant gene is that if you are heterozygous, the dominant gene is the one that gets expressed. Ergo, every single one of the F0 plants is going to come out purple, which is exactly, you might recall, the result that Mendel got, that 100% of his F1 generation came out with purple flowered pea plants. Now, let's also keep in mind here, however, that all those purple flowered pea plants, although they are just as purple as their purple parent was, they are all carriers of the white allele. They all have a copy of the recessive white gene inherited from their white flowered parents. Ergo, even though those plants are purple, they have the genetic information to make a white flower. And although they themselves are expressing the dominant condition, it is possible they could pass on the recessive condition to their offspring. Now, we like to talk about the ratios that we get in the offspring. We talk about a genotypic ratio and a phenotypic ratio. The genotypic ratio is the ratio of all the different genotypes represented. You saw that in that one, it was a 4-to-0 ratio. 4 plants come out heterozygous to 0 coming out, well, anything else. Likewise, it was a phenotypic ratio of 4-to-0. 4 to purple, 4 purple to 0 whites. All right, makes sense? All right, So that's how we build our Punnett square. That's how Mendel's first experiment worked out and why he got the results that he did. Now you might recall that Mendel continued the experiment by allowing his F1 offspring to self-pollinate. And that is going to give us a somewhat more interesting Punnett square. In the F1 generation, we are going to take the offspring from the F1 generation. These were the F1 offspring. For our F2, to get our F2 generation, we are going to cross the two F1 plants with each other. Remember, or rather, we're going to cross an F1 plant with itself. Remember that all the offspring from the F1 generation came out purple but heterozygous. So we're crossing a heterozygote with itself. This is what we call a dihybrid. I'm sorry, not dihybrid. This is what we call a heterozygote cross. Now, check out what happens here. Now we have a more interesting Punnett square because each of these organisms has one purple gene and one white gene. There's a 50-50 chance of passing on either one. So the upper right box shows us what happens if both parents pass on their dominant purple gene. You will get a purple flowered offspring that is homozygous dominance. The box on the upper left and on the lower right show us what happens. Wait, am I backwards here? Sorry, I was looking at this backwards because I'm reading through the paper. Okay, so upper left box, there we go. Upper left box is going to show us what happens when you have both parents pass on the dominant purple gene. You get a homozygous dominant purple offspring. The upper right and the lower left box show us what happens when one of the two parents passes on the dominant purple gene and the other one passes on the recessive white gene. We get heterozygous offspring just like their parents are. One purple, one white, dominant gene wins. The purple trait gets expressed. But check it out. Lower right-hand box. There's a 1 in 4 chance in this particular grouping that both parents will pass on their recessive white gene, and you will end up with an offspring that is homozygous recessive. Because that offspring does not have to compete with that white gene does not have to compete with a purple dominant gene, the white gene gets expressed. This is the only time a recessive gene gets expressed, and those offspring will come out white. That is how the white trait skipped a generation. You know, that we started off with a purple flowered and a white flowered plant. All of our F1 plants were carriers of the white gene. And then in the F2 generation, the white gene reemerges. It was there in the F1s the whole time. There's a 1 in 4 chance that those F1s, when self-pollinating, would produce a white offspring, and that is exactly what you see happening here. Now, the ratios here become interesting. If we look at our phenotypic ratio, that is, what do we actually see in the plants? The phenotypic ratio here is 3 to 1. 3 purple plants to 1 white plant, because the homozygous dominant purple offspring is just as purple as the heterozygous ones. The white gene isn't being expressed in them. So we got 3 purple to 1 white. That's what the phenotypic ratio is here. That pattern, by the way, a 3 to 1 ratio in a heterozygote cross, is so common in genetics that we've given it a special name. We call it the Mendelian ratio, after Gregor Mendel, Mendelian. But that 3 to 1 phenotypic ratio conceals a more complex genotypic ratio. My genotypic ratio for this generation is 1 to 2 to 1. That is 1 homozygous dominant plant, 2 heterozygous plants, and 1 homozygous recessive plants. So even though the homozygous dominant and the heterozygous purple plants may look the same, genetically they are not the same, because the homozygous dominant one cannot have a white offspring. It will always pass a dominant purple gene to its offspring. Therefore, every single offspring it produces is going to be purple, regardless of the genotype of the other parent. On the other hand, the heterozygotes, they have the possibility of passing on either a purple or a white gene to their offspring. And as such, there's the possibility with them that they could potentially transmit a white gene that they themselves are not expressing. They could have offspring that are either purple or white. All right, so that's how things skip a generation, folks. If you see a trait skip a generation, it is a fairly safe bet that that trait was a recessive trait, and that both parents were carriers of it, or one parent was expressing it and one parent was a carrier of it. By the way, a thing I should like to point out, and this is a really, really important thing to know. Actually, let me put a question to you all right? Let's put a question. Let's say, all right, let's suppose that you have a rare genetic disorder. Let's suppose that you discover that you are a carrier of the disorder known as sickle cell anemia. Sickle cell anemia is a blood disorder. We're going to be using it again as an example in here of another genetic phenomenon, so let's talk about it for a moment now. It's a rare blood disorder that causes a characteristic sickling or crescent shape in your red blood cells. The sickled red blood cells, red blood cells, excuse me, are not going to carry oxygen as efficiently, so you're going to be anemic for that reason. They're going to tend to clog up capillaries and other very small blood vessels, which tends to cause episodes of intense pain as circulation is cut off to whatever's downstream. You're more prone to intravascular coagulation, that is the formation of blood clots inside your blood vessels, which can make you more prone to strokes and heart attacks and things. Episodes of intense pain. You get worn out very easily when you're exercising. Your body burns through red blood cells quickly because they die very, very swiftly and have to be replenished very quickly. Most folks with sickle cell anemia do not live past their 50s, and they usually die of heart disease or some kind or another prior to that time from the strain this condition is putting on their cardiovascular system. So let us just suppose you are a carrier of the gene for sickle cell anemia. Sickle cell anemia is a recessive condition, so you've got one dominant gene for normal hemoglobin. That's the defect in sickle cell anemia, is misshapen hemoglobin proteins. Hemoglobin, you might recall, hemoglobin, is the oxygen-carrying protein in your blood. Be careful of the name of that. Do not tell me about hemogoblins on the test. Hemogoblins sound cool, blood goblins, but you do not have blood goblins. I'm sorry. All right, so, but you got one gene for healthy hemoglobin, that's the dominant condition, and you have one recessive gene for sickled, or for rather misshapen hemoglobin that causes the sickling shape in the red blood cells. That's recessive. So you're a carrier of that recessive condition. You don't have sickle cell anemia, but your kids could. Let's suppose you partner up with somebody and you decide you want to have some kids, and it turns out your partner is also a carrier of sickle cell anemia. All right. We've already seen our odds, right? You're both carriers. You know what that means. There's a 1 in 4 chance with any given offspring that you have expressing sickle cell anemia. You decide to take the chance to have some kids. And you have 1, 2, 3 kids. And none of them have sickle cell anemia. Awesome. Then you find out she's pregnant again. Does your 4th kid have sickle cell anemia? Probably not. How come? I just showed you a Punnett square that showed it's a 1 in 4 chance, right? Here's the reason why. And if you've ever been to Las Vegas, you might know this one. There's a saying in Vegas. They say at the craps table, the dice have no memory. And what that means is that every trial is a random one. It does not matter what came up on the last throw of the dice, you know. It makes no difference what happened to the previous trial because it has no bearing on the next one. Mendel's law of segregation tells us that organisms' alleles segregate independently and randomly. In other words, just because you passed on the dominant gene last time does not mean you're guaranteed to pass on the recessive gene the next time. Your body makes equal numbers of gametes of each type all the time. Ergo, yeah, there's nothing about the previous iteration that says anything about how it's going to come out now. So, in other words, what I'm showing you with a Punnett square is not this is how the offspring will come out. I am showing you the odds. It's a probability matrix, folks. That's what a Punnett square is. It is telling you the odds that any given offspring will come out a particular way. In other words, it's saying there's a 1 in 4 chance of getting a homozygous dominant offspring in that case, who does not have the sickle cell gene and is not even a carrier of it. There is a 2 in 4 chance of getting heterozygous offspring like you, who have 1 copy of the normal dominant hemoglobin gene and 1 copy of the recessive gene for sickle cell anemia. They are carriers. But there's a 1 in 4 chance that any given offspring will inherit the recessive from both of you and will therefore have sickle cell anemia. So by the numbers, the odds are in favor of any given kid not getting it. 75% chance they won't. And what happened on this kid has no bearing on what happens on the next one. It's a new roll of the dice every time. So you could have 8 kids and not a single one of them has sickle cell anemia. You could have 2 kids and both of them have sickle cell anemia. You know, it's just the way the numbers come up, right? This is telling you the odds. Average it out long enough, over a large enough number of iterations, you will end up approaching statistical averages. You know, Mendel's results came pretty close to statistical averages, but he ended up averaging the results of many trials to get them. So any given trial has no bearing on what happens on the preceding one. So keep that in mind. The Punnett square is a probability grid. It's not telling you what's going to happen. It does not allow you to predict what the next offspring is going to look like. It allows you to tell the odds that the offspring will come out either way. All right, let's throw a monkey wrench into the equation. You're Gregor Mendel's assistant, and you're helping him manage his plants. And one day, you know, you're distracted, you're smelling the pretty flowers, you're not paying as much attention as you should be to what you're doing, and you forget to label a plant. Now, you got a purple flowered pea plant sitting there. Purple's the dominant gene, right? And your heterozygotes are just as purple as your homozygotes. Looking at it, there's zero way to tell if it's purple with homozygous or purple heterozygous with a recessive white gene. So you got to solve this, or Father Mendel's going to be real unhappy with you, right? So what are you going to do? What you're going to do is you're going to do a thing called a testcross, T-E-S-T-C-R-O-S-S. A testcross is a special test that you do, a genetic crossing, to find out whether your unknown expressing a dominant phenotype is a carrier of the recessive genotype. In other words, you're testing to find out if my unknown purple plant is homozygous dominant for purple, or if it is heterozygous and therefore expressing purple. How do you do the test cross? The way you do the test cross is that you take your unknown that's expressing the dominant condition and you cross it with a known recessive. That is a known homozygous recessive. So I will take my unknown purple flowered pea plant, which might be homozygous dominant or heterozygous, and you cross it with a homozygous recessive plant. Why is that going to work? Let me show you why. Here is the outcome if the unknown is homozygous dominant. If my unknown is homozygous dominant, then when I breed it with a homozygous recessive one, I'm going to get all purple plants. 100% of my offspring will be purple because they're all getting a purple gene from one parent, they're all getting a white gene from the other. Therefore, purple is dominant, purple wins, every single offspring will come out purple. But let's suppose my unknown is a carrier of the recessive allele. If my unknown is a carrier of the recessive allele, this is the Punnett square that I get. And you see it's a little bit different, right? Because my known homozygous recessive is down the side there. My heterozygous purple parent is across the top. One dominant purple gene, one recessive white gene. You can see that in this instance, we're going to have a 50-50 chance of getting a recessive offspring. Every offspring is getting a recessive white allele from the white parents, but there's a 50-50 chance they will get the white allele or the purple allele from the unknown. If they get the purple, they'll be purple. If they get the white, they'll be white. 50-50 shot. Both genotypic and phenotypic ratio for this one is 2-2. Now, the reason the test cross is effective is because if any of the offspring, even one, shows the recessive condition, that guarantees that the unknown must be a carrier of the recessive condition. Because unless the unknown is passing on a white gene, there's no possible way for the offspring to come out white because they're all getting a purple gene from the homozygous dominant purple parent. So basically, if I do my test cross and any of my offspring show the recessive condition, I know that my unknown is a carrier. If none of the offspring show the recessive condition, does it guarantee that it's not a carrier? Not necessarily. Because remember, the dice have no memory. It is conceivable I could do my test cross, and just by luck of the draw, every single offspring could end up having inherited the purple gene. and therefore they all look purple and you think that there's no carrier in there. So it's not a guarantee. But because it's 50-50, you have enough offspring, mathematically the odds are very much in favor that one of them is going to turn up white if that recessive gene is there. And if I'm a plant producing dozens, hundreds of seeds at a time, yeah, one of those is going to show the recessive condition. It's almost uncertain they won't, as long as that recessive gene is there to be shown. So the test cross is a reasonably effective way, if you can do multiple trials, that is, of determining the genotype of the unknown. So all you need is one result to come out with the recessive condition, and you know what's going on. All right, sound good? All right, so we can also ramp this up. What I did here is I showed you a relatively simple situation. This is what we call a hybrid cross. Monohybrid, mono means one, meaning we're tracking only one trait at a time. We're just tracking what happens with a single genetic locus, like flower color in this instance. And by the way, Mendel repeated his experiment with several other traits. He repeated it with leaf shape and pea pod color and pea texture and like 7 different things he tracked, and he got similar patterns in all. Because it happened, and this worked out well for the science of genetics, but all the traits that Mendel chose to study were all traits that had a dominant and a recessive pair. Now, you can track more than one thing at a time if you want to, but it gets complicated. If we are doing a dihybrid cross, we're tracking 2 genes at a time, not just 1 gene at a time. My dihybrid becomes a bit more complex, because in my dihybrid, I have to worry about the fact that I'm keeping track of 2 different genes, each on its own locus. And Mendel tried this, because he wanted to find out if being dominant for one thing made you more or less likely to be dominant for the other thing. And so what he did is he took 2 plants that were homozygous dominant, he'd selectively bred them the same way, for 2 traits rather than 1 trait, and 2 plants that were homozygous recessive for 2 traits rather than 1 trait. In his particular exercise, he chose color of peas and texture of peas. All right, I'm just going to write some things down here to show you how this works. Now, in pea plants, the dominant condition for texture for pea plants is for them to be wrinkly, for the peas to be wrinkly. The recessive condition is for peas to be smooth. The dominant color for peas, you may be surprised to learn, is not green, it is yellow. Green is recessive. So smooth green peas, which is what we tend to expect to see in our bags of peas we get at the grocery store, are actually recessive for 2 things. So let's say that we've got it set up like this, right? That we've got, we established that wrinkly is the dominant condition, so we're going to use a big R for wrinkly. Yeah, I know that's not the best phonetics, but let's go with it. And little r for smooth. That's the recessive condition. So big R, dominant, wrinkly, little r, recessive, smooth. Likewise, we're going to use a capital Y for yellow, because yellow is the dominant condition, and a lowercase y for green, which is the recessive. All right, so when we are setting this up, it becomes a little more complex, because Mendel started off with a plant that was homozygous for both dominants. That is, one that was he knew was homozygous from selective breeding for the wrinkly condition and the yellow condition. And then he had a plant that was known to be homozygous for both recessives, that it was definitely green and smooth peed. So we are going to do a thing here where we hang on, drew that wrong. Okay, we're going to do a crossing that looks like this. We're going to cross the one that's homozygous dominant for 2 things with a thing that is homozygous recessive for 2 things. And you know what? We don't even need to draw a Punnett square to show how this is going to come out, because you can guarantee that the first organism, the one that's dominant for both, is definitely going to pass on one from, one wrinkly gene out of the 2 wrinkly genes that it's got, and it's definitely going to pass on 1 yellow gene out of the 2 yellow genes that it's got. Those are 2 different genetic loci, ergo it's definitely going to pass on 1 of each. Likewise, the recessive one for both is definitely going to pass on a smooth gene and definitely going to pass on a green gene. Those are the only options it's got. So all of our offspring in the F1 generation for this test are going to come out with that genotype you see down there. Big R, big Y, little y. They're both going to be, all the offspring in that generation are going to be wrinkly yellow peas, but they're also going to be carriers of the green and smooth genes. All right, haven't shown anything there that we didn't expect to see, right? Now Mendel is going to take those yellow wrinkly heterozygotes and allow them to self-pollinate. He's going to cross one with the other. To figure this out, we are going to need a somewhat more complicated Punnett square. We have more options to work with here, since there are more possible combinations with 2 genes in the mix. The 4-box Punnett square we did previously is not going to cut it. We are going to need a 16-box Punnett square to track a dihybrid, D-I-H-Y-B-R-I-D, dihybrid cross, which we're tracking 2 traits at once. Remember that our parent organisms are each going to pass on 1 of their 2 genes for texture and 1 of their 2 genes for color. So they're going to pass on either a big or little r and either a big or little yellow. So when I'm setting up my Punnett square, here's how it's going to look. Box Punnett square, but I've got 2 letters beside each column and 2 letters above each row, because remember, you're definitely passing on 1 color gene and 1 texture gene from each parent. So my options are, I could pass on the dominant for both, that is 1 wrinkly gene and 1 yellow gene, or I could pass on dominant for texture but not for color, wrinkly and smooth, or I could pass on dominant for color but not for texture, that is 1 smooth and 1 yellow, or I could pass on the recessive for both, wrinkly smooth. And that's the case for both parents, since we're doing a heterozygote cross, allowing the organism to self-pollinate. And let me just fill in a box or 2 to show you what this actually looks like in practical terms. The rule with a Punnett square, folks, I didn't say this before, when you're doing a Punnett square, the easy way to fill in a Punnett square is to take what is above each column and write it in all the boxes all the way down the column. And then you take what is beside each row and write it in all the boxes all the way across that row. That's your easiest way of doing this, right? So I'm just going to fill in a couple different ones here to show you ways that this could come out. Okay, I filled in 4 random ones just to show you possible ways that this could come out. Let's look at the second row, second column. You can see I passed on big R, little y from one and big R, little y from the other. Like big R, little y from beside the row, big R, little y from above the column. I ended up with a plant that was homozygous dominant for texture and homozygous recessive for color. This is a wrinkly green pea. Further down the row, I've got a plant that got big R, little y from across the row, but it got little r, little y from above the column. This plant is also wrinkly green, but the difference here is that it's a carrier of the smooth gene. Look at first column, third row. We got little r, big Y from next to the row, that is smooth and yellow. We got big R and big Y from above the column, that is wrinkly and yellow. We have a plant here that's heterozygous for texture, one wrinkly gene and one smooth gene, but homozygous dominant for color. So this is going to be a wrinkly yellow pea plant, but it's carrying a recessive gene for smooth. And then let's look down here in the last box that I filled in. That's going to be the third column, last row. We got little r, little y, that is smooth green from across the row. We got little r, big Y, that is smooth yellow from above the column. That gives us a plant that is homozygous recessive for texture. It's going to be smooth, but heterozygous for color. So we got a smooth yellow one here, but it's carrying a recessive gene for green. Now, I'm not going to fill in this entire 16-box Punnett square because it would take up the rest of this lesson, but I will tell you this. Gregor Mendel predicted what was going to happen. He believed that what was going to happen was that there would be independent segregation of the alleles. He believed that based on his previous experiments, there was nothing to think that, no reason to think that being dominant for one thing meant you more likely to be dominant for another thing, or being recessive for one thing made you more likely to be recessive for another thing. Mendel did not think that was how it was going to work. He figured that each of those genes would segregate independently of the other one, and therefore that you could have any possible combination, and it was not more likely to be dominant for both just because they were dominant, nor was it more likely to be recessive for both just because they were recessive. By the numbers, you're going to see more dominant than recessive because heterozygotes, the dominant wins, whereas recessives have to be homozygous to get it. Mendel did the math on this, and he predicted that his final result for the phenotypic ratio, what we would actually see in the offspring, would be 9 to 3 to 3 to 1. In other words, he predicted that you would end up with 9 plants that were homozygous for both. 9 plants that expressed the dominant condition for both, that is yellow wrinkly. 3 plants that were going to express dominant for texture but not for color, that is wrinkly and green. 3 plants that were going to express dominant for texture, for color but not for texture, that is smooth and yellow. And 1 plant that was going to express the recessive for both, that is smooth and green. 9 to 3 to 3 to 1. So dominant for both, dominant for texture, recessive for color, dominant for color, recessive for texture, and recessive for both. And what I want you to do, you can pause and do that right now or do it after you're done listening to the lesson, is I want you to go through and fill in that Punnett square and determine for yourself, you know, to find out the genotypes of all the organisms, count up the phenotypes, not the genotypic ratio. The genotypic ratio for this thing is going to be a mess, but count up the phenotypes, how many are dominant for both, dominant just for color, dominant just for texture, or recessive for both, and see if Mendel was right. See if his prediction was in fact correct. Okay, I'll spoil you on this. Mendel was absolutely right. There's nothing about 2 dominants that links them together. There's nothing about 2 recessive that links them together. But fill out the Punnett square anyway so you can see why. So that you can count up the numbers for yourself and see why it came out that particular way. This led to Mendel's law of independent assortment, which tells us that different genes on different loci assort independently of one another. In other words, there's nothing that's going to make gene X and gene Y both assort in the same way. There's no reason 2 dominants or 2 recessives are going to sort together that would make them likely to happen that way. All right? I realized before, when I was telling you about Mendel's results, I told you about what happened in his F1 generation. He bred his F0s, the F1s, all came out heterozygous. They all came out with the purple flowers, right? And then I told you in his F1 generation that we bred those, and in the F2 generation, we had the Mendelian ratio, 3 to 1 genotypic, 3 purple to 1 white, and in the, but we also had, that's the phenotypic ratio, what we actually see. The genotypic ratio is 1 to 2 to 1, 1 homozygous dominant, 2 heterozygotes, 1 homozygous recessive. I never addressed what happened in the F3 onward, and you might recall from earlier in this lesson when I was telling you about Mendel's F3 generation, what happened there. That in the F3 generation onward, he got pretty consistent results no matter how long he left the plants to self-fertilize. He found that 1/4 of his F2 plants were true breeding purple. They always produced purple offspring no matter how many generations he let them self-pollinate. 1/4 of his F2 plants were true breeding white. They always produced more white whenever they self-pollinated. And half of his F2 plants repeated the F2 result when they self-pollinated. You got a 3-to-1 ratio. Now you can see why that is. The one that always produced more purple was the one in the upper left, the homozygous dominant one. You let that thing self-pollinate. I don't even have to draw the Punnett square. We can predict that. It's going to be a Punnett square full of capital R's, right? You know, it's always going to make more purple offspring because there's no white gene in there to pass it on, no matter how many generations you let it go. The 2 out of 4 that repeated the F2 results are the ones that are heterozygous, because this square that I'm showing you right here, the F2 square, is also going to be the same square for the F3 if they are heterozygous, you know, because when they cross, you got a 1 in 4 chance of getting both dominants, 1 in 4 chance of both recessives, 2 in 4 chance of getting one of each. And you can see why the white are true breeding when they self-pollinate, because for crossing homozygous recessive with another homozygous recessive, no way to get a dominant gene in there to compete. Don't even have to draw that Punnett square. It's going to be full of little r's. All right, so that is how we get Mendel's results here. This is why he got the results that he got. This is the basic working of dominant versus recessive with our homozygote crosses and our heterozygote crosses. And hopefully, if you've been following along, you've been drawing some of these squares, and you can play with this for yourself. And I recommend you do that. Try some different pairings. Try crossing, you know, a homozygous recessive with a heterozygote and see what happens. Try crossing a homozygous dominant with a homozygous recessive, see what happens. Try it with a homozygous dominant and a heterozygote, see what happens. Try a lot of different combinations. Play with it and watch how things come out. And you can see you can make some pretty accurate predictions of the likely results for the offspring. Again, dice have no memory. This is telling you probabilities, not what is definitively going to happen. All right, now we were lucky. We as in geneticists were lucky that Gregor Mendel chose to do the plants that he did, chose to do the experiments that he did with them, because Mendel smartly picked paired features that only had 2 variations. And fortunately for genetic science, he picked 7 features that all had 1 dominant gene and 1 recessive gene for them. That made it easy to interpret the results and easy to deduce the pattern. It's not always that simple. I have not addressed what happens if you have a situation where you have more than 1 dominant gene, or what happens if you have no dominant genes, or what happens if you have more than 1 gene that affects a single trait, or 1 gene that affects more than 1 trait. All of these things are things that can happen in genetics, and we're even getting into the fact that there are some things that affect men and women differently. So I think this is a good point to leave this lesson off. We have heard a good little bit here. In our next lesson, which might wrap up this chapter depending on how quickly we go, I'm going to tell you about the odd phenomena. We're going to tell you about the things that are not as clear-cut as 1 nice dominant and 1 nice recessive. We're going to look at codominance. We're going to look at incomplete dominance. We're going to look at polygenism. We're going to look at pleiotropy, and we're going to look at sex linking. And that's all the kind of stranger phenomena that don't fit in quite as nicely with this pattern I have described. All right, this all sound good? I hope you have all enjoyed. Oh yeah, one last little thing I want to throw in here. I don't think I finished telling you the story last time. I told you that poor Gregor Mendel never got any recognition for his paper during his lifetime. That, you know, he presented it to like 40 people, none of whom really understood it. He published it in an obscure journal. Hardly anybody doing genetics read it. And then Mendel left science pretty much because he got caught up in the politics of the monastery he was living at and ended up running the place and didn't have time for experimentation thereafter. So how do we know Gregor Mendel's name at all? The reason we know Gregor Mendel's name at all is because in 1900, 3 separate geneticists working independently all rediscovered the same things that Gregor Mendel had found. A German geneticist named Karl Correns, C-O-R-R-E-N-S, a Dutch geneticist named Hugo Virchow, V-I-R-C-H-O-W, and not Hugo. I don't remember what his first name was. No, I was thinking Hugo de Vries. My bad, wrong person. Virchow is the correct last name, though, Virchow. And another German geneticist by the name of Erich von, we'll see his name written as Erich von Schermark, Tsechermark, or you'll see his name listed as von Sessenegg. That was actually his title. He was a baron. But Erich spelled E-R-I-C-H. Either way, in the year 1900, these three separate geneticists, Erich von Schermark, Karl Correns, and Virchow, all independently discovered the same things Mendel had discovered. All of them did their homework and poked around in the literature. It just so happened that two of the three of them were working with pea plants. They found Mendel's paper, and all three of them decided that the responsible scientific thing to do was to give credit where credit was due. There might have been a little bit of a self-serving thing in here because all of them were proposing something they figured was going to be controversial in genetics, and it was helpful to have their idea backed up by somebody else who had already done it. So they could say, here, I've confirmed the results of Gregor Mendel's research. So all three of them independently published their work. All three of them independently chose to give credit to Gregor Mendel for getting to the idea before they did, rather than claiming the spotlight solely for themselves. It burnished their results. They never got as much fame for Mendel as Mendel, ironically enough, because, frankly, we tend to hang a big ribbon on whoever comes in first and forget about everybody else. So I like to give those three their props. They were all great geneticists. Had Mendel not been around, all or any of them might have been the name I would be talking about in this class, and we might be talking about Schermarkian genetics rather than Mendelian genetics. But all three of them did the good thing. They were all responsible. They gave credit where credit was due for priority in the research. All three of them kind of slid into scientific obscurity as a consequence of that. Well, Virchow did not. He became really famous for other things in biology. But I like to give all of them credit for doing the right thing there, and just to say, hey, these guys found it too. But it is because of their doing that that we know the great name Gregor Mendel at all today. Incidentally, after that was done, it was not too long after that when an American geneticist by the name of Thomas Hunt Morgan and a British geneticist named Ronald Fisher, who was actually the great-grandson of Charles Darwin, two of them got these ideas together. They paired up Darwinian natural selection with Mendelian inheritance to create a fusion idea for explaining both heredity and evolution that was better than Mendel's or Darwin's ideas separately. They called it the modern synthesis, and the modern synthesis is the dominant paradigm still in biology to this day, although obviously we have built on it since then by learning about DNA and other things that these guys didn't know about in the early 1900s. All right, so that's it for that story. Thank you for your attention. Next time we will get to the weird phenomena. Hope you're all having a good evening. I'll talk to you soon.